Hi, good morning, everyone. As Bill said, my name's Meghan Capps. I'm currently a research assistant at the Auburn University Shellfish Lab. And today I want to talk to you about a project we've been working on looking at the performance of Gulf of Mexico tetraploid lines in different salinity environments. Just kind of as a quick outline, so I'm going to go into a little bit of why we want to look at tetraploids. We'll then go into the results and findings of this research, but I want the main take-home message to be the future directions of this work. I think it pairs well with some of the other talks you've heard this morning, and it's really exciting for the future of oyster research in the Gulf of Mexico. So starting off, what are tetraploid oysters? So tetraploid oysters are oysters with 4 sets of chromosomes within their cells. This is in contrast to diploids, which have 2 sets of chromosomes within their cells. We use these tetraploids to produce triploids. This is done by fertilizing a diploid egg with a male tetraploid sperm, thus giving us the triploids. So triploids are oysters with 3 sets of chromosomes in their cells. They are relatively reproductively sterile, and they can grow faster depending on the environmental conditions that they're grown in. They are heavily relied on in the farming industry. In the Gulf of Mexico last year, the Auburn Shellfish Lab actually produced 55 million triploid seed for the industry. So the main reason for this work is the oyster mortality events. So the first events were in the Gulf of Mexico were observed in 2016. It's important to note that these are very complex and nuanced events. It's likely a combination of multiple factors causing them. However, due to the parental genetic contribution of tetraploids to the triploids, it's important for us to assess how tetraploids respond in different environmental conditions. Which brings us into our research questions. So how is the growth and survival of current Gulf of Mexico tetraploid lines affected by different salinity environments? And what tetraploid lines survive best at each experimental location? So starting off with our broodstock lines, so we had 2 Gulf of Mexico specific tetraploid lines we use. We have 4M GNL20s and 4M2 APCK20s. So the GNLs are of Louisiana origin, the APCKs are of Florida origin. For simplicity's sake, I will be referring to these lines as Louisiana tetraploids and Florida tetraploids. So with these 2 broodstocks, we produce 2 pure lines, and then we produce 2 reciprocal crosses swapping parentage. These oysters respond in the AUS cell spawning system. We reared them in 4,000-liter tanks at our hatchery. They were set on culch and grown out in our nursery before being deployed to our farm site for further grow out prior to experimental deployment. Our study area is coastal Alabama. We have 3 sites that all experience different salinity regimes. Mobile Bay is our low salinity site. Grand Bay is our moderate salinity site, and then Dauphin Island is our high salinity site. 4 replicates of each experimental cohort were deployed to each of the 3 sites in August of 2022. Each replicate was deployed amongst one of 4 oyster grow cages. These cages had 6 slots. The middle slots were left empty to reduce any variability due to water flow. Survival and shell metrics were measured over the course of 12 months. And just for reference, anytime I refer to oyster size, I will be talking about shell height, which is the distance from the hinge of the oyster to the bill. So just quickly looking at our environmental data, so there's a lot of fluctuation in salinity at all of our sites, which is typical for the Gulf of Mexico. In Mobile Bay, we had an average salinity of 13.9. At Grand Bay, we had an average salinity of 19.8. And then Dauphin Island had an average salinity of 24.5. So jumping into our results, so here we have the average % survival at our low salinity site. The Louisiana line survived significantly worse than the other 3 lines. All lines saw below 50% survival. Here we have survival over time. So on the primary y-axis, which should be to your left, we have the average percent survival, and then the secondary axis on the right has the average daily salinity. Salinity is shown by the black line, and you can see that the greatest mortality event we saw at this site actually happened prior to the first month of sampling. The oysters were deployed during a period of low salinity, and then after that, the survival kind of stayed steady up until early spring, where we saw a little bit more mortality during an additional low salinity event, but nothing as extreme as what we saw initially. Here we have average percent survival at our moderate salinity site. So here again, all the survival was below 50%, and the Florida line actually exhibited the lowest amount of survival across the 4 lines. And then again, we're looking at the survival over time. So here, we saw kind of a gradual decrease in survival from the beginning, but then once again, during the early spring, going into early summer, there was a decrease in survival. This paired with another low salinity event, and you can see there was a large amount of fluctuation in the salinity that likely also influenced the survival as well as the low salinity. It's also important to note at this site there was a large dredging event that occurred that may have also played a role in some of the survival. And then here we have the percent survival at our high salinity site. So overall, this site saw the lowest amount of survival, but when looking at differences among the 4 lines, there was none. And then this is my favorite figure. So this one really highlights the spring mortality we saw at this site. So there was very little mortality for the first few months that these animals were deployed. And then it dropped pretty rapidly over the course of about 4 months. And even though overall the salinity was higher than what the other sites experienced, the salinity fluctuations were much greater here. And there was also a separate dredging event that also occurred here that may have played a role. And then this one, nothing super interesting here. This is kind of what we got for shell height. So at our low salinity site, Mobile Bay, the oysters grew significantly slower than at our other 2 sites, which is to be expected. And this is just kind of an aside. I really like the contrast of these 2 pictures because it shows the impact that wave energy can have on the shape of and look of an oyster. So the oyster on the left came from Grand Bay, and the oyster on the right came from Dauphin Island. Dauphin Island gets a lot more wave energy than Grand Bay. And so you can see that the oyster from Grand Bay has kind of a rough growing edge. It's a little muddier, whereas the Dauphin Island is it's clean, is really smooth, and this can also affect the growth of the oyster as that growing edge keeps getting knocked down. So for my conclusion, so at the moderate and high salinity sites, all lines experience an increase in mortality in spring through early summer. The pure Louisiana line may not be the most optimal choice for use in low salinity environments. This is contrary to what we were expecting, so it'll be interesting to see how this plays out in the future. And there were no significant differences in the survival at the high salinity site. So going into our future work, so right now we're in the process of looking at triploid populations that were bred from the tetraploids that we used in this experiment. These animals have now been at the same experimental site for 4 months, and so we'll be monitoring them the same way to see if survival mirrors what we saw with their parents. We're also planning on assessing dermo load over a course of time with these animals. We just had our first dermo sampling a couple weeks ago. We saw very few dermo cells, which is good, but to be expected considering the animals are relatively young and haven't been in the field very long. We are also working with Joey, Matt, and Chris Hollenbach at Texas A&M Corpus Christi to assess population structure and genetic variation within the current triploid experimental cohorts. And then overall, we're planning to selectively breed to produce tetraploid lines for specific salinity environments within the Gulf of Mexico. I'd like to thank the Auburn Shellfish Lab and their staff and graduate students for their help. We appreciate Foresea's technologies for the development of the Gulf tetraploid lines, and the funding was provided by the USDA. I'd be happy to take any questions.
All right, do we have any questions for Meghan?
Hi. So you mentioned that the moderate to high salinity sites, spring, early summer mortality, and you'd mentioned dredging also. Was the dredging concurrent with that spring and summer mortality? And if so, what is it about the dredging that you think might be connected?
Yeah,
to
so it happened during, so we were already kind of seeing some mortality prior to the dredging. It did happen during that time, so I think the dredging was late spring, early summer. And there was a lot of sediment in the water, so it was noticeable bringing the oysters in that there was an increase in sediment in the water. They came in muddier than they had been, especially that high salinity site. So you had that clean oyster I showed you. They weren't coming in like that during the summer. It looked like a completely different site. So,
Any other questions? Here, I'll just pass it down.
Is the Florida line of oysters being grown anywhere in Florida or tested in Florida?
sorry, what was that?
Is the Florida line of tetraploids being tested in Florida? Because the water conditions in Mobile Bay are significantly different than what you see throughout the panhandle of Florida.
Yeah, so I know we're working on getting some of these animals over to Florida and expanding the locations they're at. Yeah, yeah. So and we send the tetraploid sperm there. So
So Leslie, you want to just,
Scott provides several facilities, and including me, to hold and grow the tetraploids. I think Scott, we're on the 4th generation of the Florida tetraploid line.
yeah, is that third? I'm seeing 3 fingers, third generation.
No, Scott, I think I got to have 4. So, no, I don't know.
All right, any question here?
It seems counterintuitive from what I think about Louisiana having a lot of fresh water and Florida having a lot of salty water, although I'm sure there are places in Louisiana that are. Do you know specifically the where these where these oysters were from?
I don't. So that's something we need to go back. We've talked about going back and kind of tracing the lineage. They've been around for a little while. I'm not exactly sure how many years.
Well, I can tell you where the original Florida stocks came from. They were selected. We harvested from wild populations in Cedar Key, generally a high salinity site, and then Apalachicola Bay.
Thank you.
All right, with that, we're going to move on. Thank you, Meghan.
Yeah, absolutely.